Okay, so first I want to emphasize that this work was done with two people in my group, Ian Hepburn and Wei-Lang Chin, and also in collaboration with the Human Brain Project at EPFL, who helping us with basically kind of the software engineering of these things. So my talk will be split into three parts. So I'm first going to do a bit of biology and really discuss work we actually did a few years ago, which shows why we're really interested in parallelizing this inhomogeneous SSA process. Then I'll go through kind of a more theoretical part where I show the operator split method which we've chosen and developed, and demonstrates accuracy in comparison with other methods, and already show kind of an initial parallelization. And then finally, this will be more like kind of a computer science part, where we really then optimize the method and show how we apply it to a full cell model. And so let's start with the biology. So I'm basically a computational neuroscientist, so we're interested in neurons. And first of all, neurons have these very complex shapes, and so we tend to be really interested in modeling morphology in detail, which is why our method uses tetrahedral meshes, so that we actually can capture this morphology in full detail. And one of the phenomena we're interested in is this electrophysiological behavior in the dendrites of neurons. In this case, these are Purkinje cells. They're one of the main neurons in the cerebellum, which is in the back of your brain and involved in motor coordination. And these cells show something called calcium burst, where basically you get a large voltage-gated calcium influx into the membrane, which depolarizes the membrane. And what we noticed in these recordings which were taken in cerebellar slices is that actually if you look at these bursts, and here they're blown up, that they show a large variability in shape, in the number of spikes in the burst, and so from burst to burst. And this is it's a very simple experiment, so it's current injection here in the soma. So, and this variability has been described a long time ago, but within the community there was not really a very good explanation of what was causing this variability. Right? And so the hypothesis we're going to pursue is that it's actually stochasticity of the gating of the ionic channels involved in causing this bursting behavior. And so we explored this so we've been developing this software called STEPS, which basically does the spatial Gillespie on tetrahedral meshes. And but to be able to model something like that, you also need to compute the membrane potential, which is something that does not really fit that well within a Gillespie context. So we have this separate process which computes the membrane potential on the same mesh. Which I think is a real advantage, that you use the same mesh for both. And then all the rest, so we're going to have calcium diffusion and so on, but also gating of ionic channels in the membrane is in the Gillespie part. And they basically communicate with each other, and that works quite well. And then to look at this stochastic effects, we basically looked at a simple cylinder, and we changed the length of this cylinder to go from kind of a small system to a large system. And if you do that doing the standard Hodgkin-Huxley model of a spike, we basically recapitulate something that's well known in the literature, namely that if you look at a small system for the spike, there is a lot of extra spikes, so you get spontaneous spiking in this system in the small system case. And also a lot of jitter in timing of these spikes. And as you kind of make the cylinder longer and longer, and so go to the large system case, basically this stochastic behavior disappears. Right? So we just replicate something that's known in the literature for gating of voltage-gated channels. And that's the important point here. The classic Hodgkin-Huxley is a purely voltage-gated system. For this dendritic calcium spike model, however, this is no longer purely voltage-gated. So this is the model we basically use. It has 4 different types of membrane channels. So there are 2 types of calcium channels and 2 types of calcium-activated potassium channels. And then there is a calcium influx, and there's a whole buffered calcium diffusion process, pumps. So there's a lot of stuff going on. But in particular, you have these channels which are activated by the calcium concentration. In this BK channel is also sensitive to the membrane potential. But so there's a calcium activated process here for activation of some of these channels. And so if you do then the same thing as we did before, so you basically look at the proper, and this system is kind of set up so that it will generate a spontaneous calcium burst, and that's just done by having enough of this T-type calcium channel in there. And so if you now look at the same at this in stochastic simulation, you see that you get extremely noisy system both in the small system case, but also still in the large system case. And in particular, in this longest cylinder, you still get a huge variability in the number of spikes that's being generated, and they're color-coded, so it's easy to see. And when you align on the first spike, it's even more obvious. So basically, when you introduce this calcium activation step, for some of the channels, the stochasticity of the system is persistent and stays also for large systems. Right? So that's really different from what you see in the purely voltage-gated case. Now these cylinders are kind of now, and then in the paper we went in and basically used combined stochastic deterministic modeling methods to show that it's really this calcium activation step which is the important component in this behavior. But these cylinders are not real neurons, they're kind of an abstraction, so it would be nice to look at the same behavior in dendrites, and we did that. And so that's what you now see. So this is something slightly different now. Now we have basically modeling a synaptic response, something called a complex spike. And what you see now, in addition to the variability in the temporal domain, where sometimes you have 3 spikes and sometimes just 2 spikes in this complex spike, you also see a really large spatial variability. And that's something we didn't really see before in these cylinders. So you get now both temporal and spatial variability in this stochastic system. So that's very nice, but there's one little ad hoc in these simulations. So we only really could simulate a small part of this dendrite, and that took us 3 weeks to compute. Right? So some people have been remarking on how long their simulations lasted. So this is among the worst cases, I think, reported in this workshop. And so obviously, and this is because we're doing this on a single processor. Right? So this is not parallelized. So we want to parallelize the software, and in general also, so STEPS is really a program which is open source, distributed, and we try to make this kind of foolproof. Right? So a lot of people who are interested in this modeling are not really that sophisticated, and so we want to have something that's very robust. So the spatial Gillespie or inhomogeneous SSA is inherently serial, that's well known. So this is again here kind of replicated. So if you try to parallelize it as it is, you can get kind of an increase in speed of 2 to 3, but that's the max. And this basically because as these different processors compute, some go ahead too far and get a message, oh, you need to go back in time. It's called rollback, and a rollback process is really very expensive in computing, and so you're spending all your time based on these rollback processes. So the way around that is to basically split up what you're doing and basically decomposing two separate processes. So you have the standard SSA, which will compute the chemical reactions, but the diffusion you're going to do differently. You basically are not going to keep track of each separate diffusion step, but you're going to combine them over some time period which we call tau. And what's really important in doing this is to actually do this for multi-molecule diffusion. So it's fairly simple to think about this if you allow only one molecule to diffuse, but then your tau is going to be very small and your method is not going to be very efficient. So you want to allow for multiple molecules to diffuse. In the case of the tetrahedron, you have 4 phases, so they can diffuse over any of these phases. And what's also important to realize is that this tau really represents a time period that includes both past and future diffusion events. So that's all simple, but then you have to think about, there's a lot of technicalities that come in. So first of all, how many of these molecules are going to diffuse at tau? How do you compute tau? How do you distribute them? So there's a lot of little details you need to solve. There's quite a bit of literature on these methods already. So people have proposed different solutions. Let's go through them and compare things. So the first thing is really, what are you going to take for tau? So this is, in a tetrahedral mesh, fairly complicated, because if you just do a regular grid, it's the same on every grid place. Your local diffusion rates are the same everywhere. Here your local diffusion rates depend really on the shape and the size of the tetrahedra. Right? And so you can compute this. So they will be different for the different phases of the tetrahedron. So you can find these diffusion rates. You take 1 over the diffusion rate. Basically plot the distribution across the whole mesh, and so you see that this is very strongly peaked. There are only a few outliers there, and we find that if we take like the lower 10%, a cutoff at 0.1, that this really gives us actually a very good approximation. So this is a comparison of the probability density function for diffusion using either the standard ISA or our operator split, taking a zeta of 0.1, and this is basically an almost perfect match. So and the advantage is that if your diffusion rate doesn't change during simulation time, you basically can compute this at startup, and so just your tau becomes fixed. Right? So that's one of the advantages of the method. It basically, the accuracy is set at the startup of the simulation, and you no longer need to check during runtime. That's a big advantage. Then this is maybe relatively trivial, but still looking at what's out there in the literature, I think it's an important point. So really, if you just think about one direction of diffusion, it really should be modeled as a binomial. If you have multiple directions, it's a multinomial. And this just shows that if you actually do this, you get a very good match to the real process. Again, it's a comparison with the ISA, the sign is our method. If you just take kind of a mean instead of as it's a uniform function instead of a binomial, it really doesn't work very good for large molecular densities. In the literature, it has sometimes also been proposed to make this more efficient, to take the net, so the flux, instead of actually the diffusion. So the net diffusion, this really doesn't work very well either when you have large densities. But that's kind of not really so exciting. It becomes a bit more difficult. So this was now just pure diffusion. Now I want to combine this with the SSA, right? So because of chemical reactions, the number of molecules in your tetrahedron can change during this time step over which you're going to estimate how many molecules are going to diffuse in or out. Right? And how do you actually keep track of that? And so we've come up with kind of a very trivial approach, but which hasn't been described before. You just basically kind of integrate these changes over time and take the mean. Right? So you actually keep track of how your SSA changes the number of molecules. You just integrate this, take the mean. And that works very well. It's much better than, for example, taking the number of molecules at the end at tau itself, which can be really strongly, so this is assuming there are jumps like this, or do like, for example, median assumption also, which gives you obviously less accurate results. Yes?
This is then not splitting in the traditional sense. You're saying you're taking the reaction into account when you do the diffusion.
Yes.
It's not splitting, you do one, then you do the other.
So what we...
In the standard Lie or Strang splitting, it's not like that.
Well, maybe we can discuss about this later. So let's compare this with other published split methods. So here we basically have a simulation where you have constant influx at both borders, and so initially no molecules in the center, and it slowly builds up. And we compare here our methods with, again, the standard ISA, and then different approaches, which basically standard kind of operator splitter approaches, either first order or second order. And you see that it's kind of difficult to see here, but we're slightly more accurate than those other methods. The differences are not always that large, but you can look at this more systematically on different types of meshes, and especially compared to the first order method, we're considerably more accurate. The second order, it's slightly more accurate to comparable depending on the methods you actually look at, and these are traces here in what happens in the center, and the percent of error compared against to the ISA, and as you can see, generally speaking, the sine curve is better than these other curves. So we think we've made a slight improvement over the previously described splitting methods, and but let's now look at accuracy in more detail. So so for this TIPS simulator, we've kind of published a set of standard validation models we use. So one is a production degradation model, which has a second order reaction in it. Others kind of a multi-reaction diffusion model. They're all highly artificial models. And this just shows you that we get really excellent accuracy when you look when we test the method with these models. And in general, at that level, it's really difficult to show any difference with the original ISA, so which is nice. But as I told you, these are highly artificial models. So what's the case for a more biologically inspired model? And so we take the classic E. coli min D model, which has been used a lot as kind of a test model for spatial stochastic methods, and so I'm going to show here two different comparisons. So in the first one, we basically take the model at a particular state and then advance it for 100 milliseconds. either using the standard ISA or our operator split, and track the errors, the difference between those two over that time period of 100 milliseconds, and we do that tens of thousands of times. I've forgotten how many simulations this really are, but it's quite a large number. And so we can then compute the percent error compared to the standard ISA for the mean concentration, or and also for the standard deviation. And you see that for both of these, the error is always smaller than 0.5%, and actually for the mean it's smaller than 0.2%. So this is tracking the behavior of the system over a short time period, and basically it matches extremely well. Over longer time periods, not entirely surprisingly, the two will start to diverge. But the main behavior is still identical, and that's from a biological viewpoint quite important. So this is kind of, so this system oscillates. This is kind of represents the frequency, let's say, and this is the peak of these, the amplitudes of these peaks. And so in both cases, so the comparison, basically there's no difference between these measurements. So that's nice. So the method seems to be really quite accurate and robust for different types of models. But does it actually help with parallelization? Because that's the whole goal of this project, right? So here we now use this, again, this mean D model on for a low-level parallelization, and I haven't really emphasized this before. So what we're going to do is run this on clusters using NPI. Right? So this is kind of something that's available in most of the university computing departments. And so for this model, we just went over a relatively small number of processes. But as you, so this always plots the speed of these graphs. So you can see that we get kind of quite nice speed ups, which depend on basically both the model size and the configuration. So the model size here is basically the number of molecules you have in there. And you see that there is here a sweet spot for this medium density model. So medium density of molecules, while for lower densities, really you only get the speed up to a lower number of processes. And the high densities, the speed up is not as large for the medium density. Also the mesh affects things, and then whether how you actually what the model looks like, whether it looks more like a 1D system, a 2D system, or a 3D system. Right? And so that already hints on you. So in a 1D system, the NPI communication is going to be very simple. As you go towards 2D, 3D, it becomes more complicated, and you see that this then actually reduces the scalability. So that already hints that all these details are going to matter if you really want to have optimal scaling. So let me show you the conclusions up till now. So if you want to do spatial Gillespie, you really need to do operator splitting to get a scalable solution. You need to do this binomial correction, multinomial in the case of multiple directions of diffusion. We get this, we use the mean number of molecules to take account, change the molecule number during this tau, and that gives us good accuracy, and we see a reasonable scaling in a first small model parallel implementation. But we really want to go for these full neuron models, right? So that's and here I can come to the last part of the talk, where we now going to really test this approach on full cells and see what we can do to get good scaling there. So there are a number of important points there. So this is basically at this point a fixed size problem. So we not changing the biochemical model or the morphologies. So we use pre-generated meshes, and you also will have a fixed partitioning of the mesh. But we have this complicated dendritic tree with many branches, and that will be an important point, as we'll see. And we can have strong gradients. So what we want is strong scalability. We want to look at the partitioning and potentially at dynamic load balancing. So what does this actually look like if you on different processors and the communication part? So in essence, you will have the SSA running over this time period tau. Then we apply the diffusion operator. Then you need to have this MPI communication phase, which then communicates the diffusion cost changes in molecule numbers across different nodes in the simulation. And then potentially you need to wait sometimes till this is all finished, and during that time this process might not be doing anything. So in essence, the computation time on a single process really consists of three parts. You have the computation of this SSA and diffusion, then you have this MPI communication part, and then potentially you're just waiting. Right? So to look at in more detail at this, we've first looked at a really small model, just 55,000 molecules, and on the order of 3,000 tetrahedrons. And basically this is what it looks like. So you have, as you increase the number of processes, you have a very sharp drop in the computation time. And notice that there are two phases in this. Initially, this really drops very fast, and then you have this kind of slower drop. I'll come back to that in the next slide. The communication time decreases slowly, and this idle time actually slowly increases. It's difficult to see. But so most of the speed up comes from this reduced computation time, which is not really surprising. And these other two, except for this initial drop in communication time, kind of evolve much more slowly, which means that there is kind of an optimum size of the simulation in which you will get maximum speed up, and beyond that size, really, there is not really that much of a change anymore. Right? So again, depending on your model and what you're actually doing, there is an optimal number of processes over which you can distribute this. Now, why does the computation time drop so sharply in the beginning? This is really because modern processes are very dependent on cache memory. Right? And so as we distribute a model over different processes, on each process the model becomes smaller and smaller, and so we make more efficient use of this cache memory. And it's a very, from computer science viewpoint, a very trivial result, but actually gives you a tremendous speed up. So just decreasing the model size on each of these different processes. And so this is the decrease in memory use per processor as we go from a single to more processors. We see this initially very sharp drop, and that explains this initial very strong increase in speed, actually. Now, for communication time, so if you, and I'll come back in a minute how we do this for more complex meshes, but initially it's a very simple mesh. So if you partition things efficiently, you're only going to have kind of local communication, so with neighboring processes and no distant communication, and that really helps a lot. But then there are additionally small things you can do, like initially we kind of naively communicated all of these diffusion events, and now we kind of aggregate that information much more so that the amount of information we need to communicate is much smaller, and that led to a serious drop in communication time. So then the kind of more limiting process is now this idle time, so we want to decrease that also. So to decrease this idle time, we basically now let processors run ahead. So once they've received diffusion information from their neighbors, they know they have all the information they need to continue. There might still be MPI processes going on in other parts of the cluster, but we don't care about that, and we allow this guy already to restart, and that basically gives us a reduction in overall idle time. So again, things become a bit faster. So now, and so you see, as we make these changes now, actually the number of processors over which we can parallelize the system increases, right? Because we kind of reduce the effect of these relatively constant processes, and so this is kind of the performance increases we've been getting using these processes, and this is still ongoing, right? So the point is there's actually a lot of little fine-tuning you can do to increase this. But this was on a kind of dummy small system. So in the last part of the talk, I want to go now actually to the full branched morphology. So first of all, it's actually not really that trivial to generate a tetrahedral mesh for such a large branched morphology. So for that we use a program called Trellis, and which allows us to convert the kind of reconstructions you find in the literature into these tetrahedral meshes. So this was the mesh we used before. This is now the mesh we're going to be using for this part. So this was 15%. This is kind of the full cell without the cell body. We didn't really have a good reconstruction of the cell body, and as this is still just kind of testing the case, it didn't really matter that much. Now, how do you partition such a complex branched structure? Right? So the kind of standard approach people would say is, oh, you just put a grid on this, right? So you do kind of grid partitioning. But if you do this, you end up with this, like, look at this, what color is that? Some kind of pinkish color. At this guy here, so you have parts of these branchlets here and other parts of these branchlets here, because they've fallen this kind of right corner of the grid. Right? But these are not really connected with each other. So that's obviously not going to be very efficient. So instead you need to come up with a method which actually makes use of the topology of this dendritic tree, and which partitions this, taking into account the defined topology of these branches, and which keeps branches together as much as possible, basically. So if we do that, we actually now get this very nice, and this is using this initial version of the method which I mentioned. So this is not including all these more recent improvements we've done. Basically, because we kind of, our computing department doesn't like us to do this too often, because we're taking up a lot of cluster resources for this. But so we get a very nice speed up, very strong scalability. And so again, this scalability is supralinear because of this improve in use of cache memory. And so it's kind of factor 2000 speed up for 1500 processes. And it's clear that for this full dendrite model, we haven't reached the sweet spot yet, right? So we expect that if we go over even more processes, we will keep this supralinear increase. We don't know at this point what the maximum is where it will start to drop off. So conclusions. So we, for the second part, so we get the supralinear scaling also for large models, but there is extensive optimization required, and that is really on multiple little aspects of the program, and it's clearly not finished, right? So one thing, so I didn't mention this explicitly, but the models we're using now do not actually include voltage-gated processes. So we were not computing the membrane potential on this. So you need to parallelize this computation of the membrane potential also. We've done this for small scale, but the really tricky thing is to integrate these two together in a way that minimizes overall communication time. And that's going to take us some time to really find an optimal way of doing this. So we work on that, and the EPFL people are really helpful with that. Then we have a separate project where we're actually looking at using GPUs in combination with the MPR. So that's early phase. And then obviously longer term, we want to look also in dynamic load balancing, where actually this mesh partitioning is not constant, but is adapted to kind of the local concentration gradients in the system. And that's, again, something in which the EPFL people have a lot of experience. And that's basically it. Thank you.
So did I understand correctly, so that you tried something where you only do compute net flux, net transfer?
No, it's not net flux. Oh, we tried that, yes, sorry, yeah. But not in our method. Yeah.
So, and you commented that that doesn't work. So if you do net flux, then you have to add to it a stochastic component. That we know from fluctuating hydrodynamics. So if you can design methods that compute the mean net flux, but then you have to add on top of it a stochastic component, and that does reproduce the correct fluctuations, especially for large populations, which is where you said it wouldn't work. So that's, I mean, we do that in our codes. Of course, that doesn't work very well when you have very small populations, but it allows you to take a PDE code. If you had a PDE code on a tetrahedral mesh that was a finite volume code, you can add fluctuations to that code just by adding a stochastic piece to the fluxes in the spirit of fluctuating hydrodynamics. And we have tried it, and that does work.
But in the small molecule case, having to add that, what's basically the cost of having to add this stochastic operator?
Usually it's, I mean, generally in random numbers is not the dominant cost. It's memory traffic. So you're already computing fluxes, looking at neighboring cells. That's what dominates the cost. But it doesn't work very well for small populations. So that's
Did you consider any strategies to adaptively control the time step in your simulation?
So we actually think it's an advantage that we don't have to do this, because obviously if you have to do this, you have to kind of continuously compute some error measure, which is going to be local. So that's not something you can compute globally. And so that's really going to add a cost to the overall computation time. And so being able to actually keep this constant and not having to track this, we think is a big advantage of this approach. It might become limiting if we want to start doing more fancy things where we actually go more hybrid methods and so on. Then there might be an issue there. But at least for this type of approach, that works very well, we think.
Maybe I misunderstood. There is something counterintuitive. Typically, communication cost grows with the number of processors. So maybe you mean something else.
Yeah, no, that's because we try to keep all the communication only to neighboring processes, right? So if it's only neighbor to neighbor, then it stays local. It doesn't grow with the total number of processes.
Well, it is a network.
Yes, but that's where this point comes in. That's really important to be very careful how you partition the model. Right? So if you use this grid-based approach, then it's no longer neighbor to neighbor. Now some of them become distal, and then your communication time suffers.
So would it be fair to say that if the network had much bigger different degree distribution, and you were connecting with 100 other things, then maybe this would not be as... I'm wondering if you can say in some sense, what is the feature of the network that makes it work so well?
I think in essence the local communication. That's what and so it will I think it will become, that will disappear if you would keep track of what's happening in the extracellular medium. Right? So then
Any loops?
Yeah.
If it
Yes,
has loops,
yes,
that would be a
yes.
problem.
And if you would add the extracellular medium, that would be the case. In these structures, there's never a loop, right? So
that's it.
yeah, so that's a property of these morphologies.
I still cannot see how it can decrease. If you have one processor, there is no communication. Everything is inside. If you
That has to do with how much you need to communicate. Right, so obviously, if you just split it in two, then there is a lot of tetrahedrons which have neighbors on the other process. So you need to communicate a lot of these diffusion events. As you have more processes, the amount of stuff you need to communicate becomes smaller and smaller. And that basically reduces the communication time.
So we don't mean the same thing. I mean things that are not in the memory of the same processor that you need to communicate between.
Yeah, but that's basically the case, right? So if you, I can use it with this. This is also a Perkins cell, right? So if you kind of it's kind of difficult to see how you would partition this in two. But in general, you need to. It just has to do with. It's not only the kind of connectivity of the MPI, but the size of the buffer disk you need to transmit of the MPI, which is important. And that buffer size becomes smaller as you have more processes. And because of that, the communication time decreases.
If I understood correctly, you plan to use the same grid for electrical properties.
Yes.
And would it be advantageous to use a coarser mesh when possible?
Yes and no. So the reason we actually like to do this is that, and also one of the reasons why we really like tetrahedral meshes and not cubic grids and so on, is that from an accuracy viewpoint, it's really very important. So these membrane currents basically are very dependent on the surface you model, right? And so you want to be absolutely correct in how you represent the surface. Conversely, these diffusion processes are very sensitive to volumes, right? So you also want to be correct in volumes. And so that's so some other people in our field have kind of taken the approach that the standard type of electrical modeling of this just uses cylinders. And to kind of do the membrane potential calculation on a cylinder and then the diffusion on a more complex mesh. And basically it's very difficult to have the same surface and volume for the cylinder compared to the mesh. Right? So by just doing it on the same mesh, that problem completely disappears and you have kind of perfect accuracy at that level. But I agree, the mesh is kind of too fine for really potential calculations, and indeed that's one of the things that the EPFL people want to look at, is to use coarser meshes for the membrane potential calculation, and so that's on the list of things to do.